When microbes invade our body, we see that B cells are capable of using their antigen receptors and binding to the antigen of the microbe directly and start reading it to start their activation process. Recall, this is not something that T cells can do. T cells can only identify MHC antigen complex. However, B cells can directly bind to the exogenous antigen and read it. Exogenous antigen refers to the antigen of the microbes that we have outside our cells in our body fluid. Also, we know that B cells can act as antigen presenting cells. So it's not surprising that here I can see this B cell already build MHC-II antigen complex. And now imagine, what if this B cell present the MHC-II antigen complex to a helper T cell, and then when helper T cell becomes active, in return, it provides co-stimulator for the activation and proliferation of this B cell. And we know that several of the cytokines that active helper T cells release can co-stimulate activation and proliferation of B cells, but the major one is interleukin-2. So now that this B cell directly attached to the exogenous antigen, read and identified it, and also received the co-stimulator, interleukin-2, is ready to start proliferating. We need this cell to divide rapidly because one activated B cell cannot provide us enough numbers of antibody molecules to fight so many of this particular microbe that are flowing in our body fluids. Activated B cell that proliferates eventually give us an army of two groups of cells. Plasma cells are the ones that release antibody molecules into lymph or blood. And by the way, please recall that the lymph eventually gets into blood. And then antibody molecules through, for example, blood plasma travel to the infected tissues to target the microbes that are outside our cells. We cannot keep these plasma cells for a very long time, so eventually we lose them. However, we keep the other group, which would be the memory B cells. If this is the first exposure to this microbe, we see that these memory B cells are not going to release antibody. In general, we can say that memory B cells are not getting involved in the battle against the microbe this time, and that's the main reason they survive, and we use them in the future. But before I talk about what these memory B cells can do in the future, let's learn about a few strategies that antibody molecules use to help the destruction and removal of the microbes that are currently present in our body fluid. Please note that plasma cells, when we say they are involved in the battle, doesn't mean that they go and actually fight the microbe. Plasma cells are involved in the battle by releasing antibody, and that would be the molecule of antibody that actually causes the destruction of these microbes. For example, here I can see one antibody molecule that is attached to the antigens of two microbes. So quickly, from mathematical point of view, I see the advantage. I'm using one molecule of antibody to fight two microbes. And then we notice that when antibody binds to the antigen of some bacteria, it actually neutralizes the toxic substances that those bacteria release. And that helps to reduce the amount of damage that the bacteria toxin actually cause in our tissues. Also, when antibodies bind to the antigens present on the viruses, we see that it makes it very difficult for viruses to attach to our cells and invade our cells. Recall that viruses, to replicate, need to get inside our cells and make the cells their own host. So when antibody makes it difficult for viruses to get inside our cells, it simply means that antibody successfully reduced the viral replication. Also, antibodies attract phagocytes. Our major phagocytes are neutrophils and macrophages, and when antibody attaches to the antigens of microbes, that would be a sign for phagocytes that these are the cells that they need to, for example, phagocytize. So when the first responder, neutrophil, gets to the infected tissue, it is attracted to antibody, and in this case, the neutrophil can actually take a bite and take all these three together in. For example, the two bacteria and the antibody that is attached to them. So that would be one bite. Next, we notice that when we have complexes made of antigen and antibody attached together, these complexes actually start activating the plasma proteins referred to as complement proteins. And we know that when activated complement proteins speed up and enhance many of our immune responses. In the next image, I show you 4 antibody molecules. At the end of their heavy chains, they are attached to one another. And as you see, each one of them is attached to the antigens of, for example, 2 bacteria. So first glance, I can see that a very large bite is ready for phagocytes. Recall that antibodies also attract phagocytes. And when phagocytes get here, instead of one by one eating these bacteria, they can actually take all these together as one bite, which that saves us time. And also, I know that when antibodies behave like this, they cause agglutination of those microbes. Clumping of the microbe happen. And when antibodies bind to the antigens of the microbes, in some cases, they actually cause damage to the wall of microbes. And as soon as damage to the wall of microbe happen, the extracellular fluid can flow in and causes the death of the microbe. And clearly, a dead microbe can be phagocytized much faster than a live microbe that keep moving around. Speaking of helping the phagocytes to clean up the area and phagocytize the microbes, in the next image I can see a very smart strategy that antibodies use. Antibodies bind to the antigens that are present on organelles such as flagella and cilia that some bacteria use to move. And when antibody molecules bind to the antigens present on flagella or cilia, clearly those bacteria are immobilized. And please recall, again, antibodies attract phagocytes. So now we have a paralyzed bacterium here, and that would be a piece of cake for phagocytes to phagocytize it, and that saves us plenty of time because we do not need to chase the microbe. It's just immobilized right there, and it could be phagocytized very quickly. So these are different techniques that antibody molecules use. However, as you notice, they are all working the best against the microbes that are free in our body fluid. And now we can get back to the memory cells that we keep after this battle is over. So these are a few memory cells that we have in our secondary lymphatic organs and tissues, for example. When in the future, for the second time, we are exposed to the exact same microbe, the difference this time is that we have cells that they have experienced meeting that microbe before. These memory B cells are much faster compared to the first time to read the exogenous antigens and start their activation and proliferation process. Building an army of new plasma cells and new memory B cells happen in a much shorter period of time, and the amount of antibody molecules that they release within a very short time is more significant than the first time. And clearly we learned a few techniques that antibodies use to destroy these microbes very rapidly before the microbes get a chance to spread to our other tissues and cause a severe infection. As always, we lose the effector cells and we keep the memory cells for future use. And when we build these memory cells, that would be an indication that we have built some immunity against this particular microbe, because each time after the first exposure to the microbe, our immune response building new effector cells and new memory cells is going to happen fast, and the immune response will be more efficient.